the building and Zagaya, the organization that is hosting the Bay Area Malaria Meeting tonight, something we like to do about once a quarter. We started doing the Bay Area Malaria Meeting after, I would say, the tremendous success of our World Malaria Day meetings annually, and a request from the community to get together and share information a little bit more often. So I'm delighted to tell you we have 3 talks tonight, spanning some of the breadth of research in the fight against malaria. We have a talk in drugs, we have a talk in diagnostics, and we have a talk in new research tools. I hope to learn a lot tonight. I'm a student of the Malaria Challenge. I came to it from my experiences at Amherst, trying to persuade yeast to make artemisinin anti-malarial drugs, which also the Artemisinin Project launched the nonprofit Zagaya that is your host tonight. The mission of Zagaya is to ensure that technology that has been developed to fight malaria is deployed for maximum impact. And I think part of that is building a cross-disciplinary community, much like the one we have tonight, and communicating about our research results, looking for opportunities to deploy these technologies and tools in the fight for malaria. So, without further ado, I'd like to first introduce our first speaker, Ebere, which this is a bit of a misty moment for me because Ebere used to work at Amherst. It was tough to see her off to graduate school, but happy to see her go to Phil's lab and start doing some great research. Ebere's going to talk about her project, looking at the mechanisms of action of different scaffolds and new drugs in the fight against the malaria parasite. So,
thank you. Thanks for the introduction. Like you said, my name is Ebere, and I'm a 4th-year grad student, 5th-year grad student in Philip Rosenthal's lab. I'm a UC Berkeley School of Public Health PhD candidate. And our lab is mostly known for epidemiology, characterizing the epidemiology of malaria in the field, most especially in Africa. So, but prior to that, Phil's lab used to do a lot of work on just biochemical analysis of drug targets, including cysteine protease, way back in the day. And so this is kind of like coming back to what the lab used to do, and looking into trying to identify the mechanism of action of, in this case, 2 new novel benzoxaborole compounds against plasmodium falciparum. How many people here, like, I'm going to apologize first. I think I'm going to go over the time. And I hope there are no shotguns from, like, in WMD to shoot me down or whatever. Okay, great. But I'm assuming everyone here is pretty much in the malaria field, so I'm going to try not to dwell too much on the very basic information here. So just going into the parasite life cycle, I'm going to be really focusing here in the red blood cell stage. That's kind of fuzzy. But this is a 48-hour life cycle for plasmodium falciparum, and involves merozoites invading our red blood cells and going from rings to trophozoites to schizones in a 48-hour cycle. And a lot of the disease that we see with malaria is due to this cycle. Hence, a lot of anti-malarial drugs tend to target the inhibition of what's going on in this cycle. However, there's been a demand for new anti-malarial compounds that most likely have potential to target different stages of the malaria cycle. So what do we know in terms of therapy? There are quite a whole number of, this is just a handful of the treatments that we have against malaria currently, and so most of it already we have a resistance to in plasmodium falciparum. And the first line mode of treatment for uncomplicated malaria currently is ACTs, known as artemisinin combination therapies. However, studies have shown that we're beginning to have resistance to artemisinin monotherapy in some parts of Southeast Asia. And looking back in history, that's usually the trend, where you begin to have resistance coming from that region of the world and pretty much makes its way to the rest of the world. And so this is not good. So there is a call for new anti-malarial compounds with new and novel mechanism of actions. And so this here is a slide from MMV, just kind of trying to give you an idea of like the different things that are going on as far as drug malaria research is concerned, and different ones at different stages. I'll be focusing on oxaboroles, which is synthesized by this company called Anacor in Palo Alto, and they're boron-based compounds. The general structures of oxaboroles are characterized by this oxaborole ring, and this benzoxaborole name is derived from the fact that it has a benzene ring attached to it, and each of these different positions can have different R groups. So what's special about boron? I get that question a lot, like what's so special about these boron compounds? Well, one of them is that boron has an MTP orbital, which makes it an excellent electrophile compared to carbon, which means that a nucleophile is more likely to attack boron compared to carbon, forming a covalent bond. And the good thing, a lot of compounds in general, a lot of antimalarials that we have out there interact with their target either by hydrophobic interactions or by hydrogen bonding. So this covalent bond is advantageous not only because of the fact that it's a stronger bond, but also it's reversible, which means that you're not going to get the whole superantigen phenotype going on. But the fact that it's such a high, it's an excellent electrophile means that the hydroxyl group in water, which is all over in our body, can be an excellent nucleophile to attack, for example, to attack the boron again, releasing this covalent bond. So this is an example that I got from a review that was published by scientists at Anacor, showing how a molecule, one of their molecules, could be interacting with the target. And so the good thing is, work has been done showing that it's a slow, this reversible covalent bond is slow in the sense that eventually you begin to get a release of your compound from the target. And another thing is that the boron is essential. Okay, now it's beginning to look funny, I guess because of the Mac.
Oh.
Yeah, well, so the good thing is that the boron is essential for activity. So what's supposed to be here, I use the Mac and this is a PC, so that's why some of this is not showing up all. But one of the things, what you're supposed to be finding here is this compound called AN2690 that was published in Science Journal in 2007 to show, it was shown to have activity against lucyltyrrhenate synthetase. And this compound, when you have boron, here you have an IC50 of 2.1 micromolar, but when you remove the boron here, it's supposed to be here, and add a C, a carbon here, it goes, you lose activity practically, and it goes to more than 100 micromolar. So, just to give you an idea of some of the compounds that I'm going to be talking about today, one is this compound 1467, and the other is compound 3661. These really were used as controls. So compound 1467 and compound 1466 are pretty much alike, except that this one is an enantiomer of this one, and this is the active compound. This is the inactive compound. This compound here, 1474, is a relative of 1467. The only difference is that this has a boron and that has a chloro group here. Sorry, a bromo group versus the chloro group. And this is the negative enantiomer for this compound. These two belong to the same scaffold and are expected to act the same because they both end with this carboxylic group here. That's really all you need to get from here. And my project is focusing on the highlighted compounds. So when I started the project, I started, the compound of interest was the 3664 compound. And our hypothesis was that this compound inhibited lucyltyrrhenate synthetase. And the reason why we chose that was because I needed to pass my quals one. And at the time, the publication in Science in 2007 was really the only published information we knew about how the benzoxaboroles work. And that showed to have activity against yeast fungal URS. But outside of that, more than that, there's we also came to that hypothesis because structure-activity relationship was done for 3661 and was shown to be similar to the AN 2690. And so it seemed like a good place for us to start, really. And later on, publications came out concerning the activity of this benzoxaborole against bacterial lures. 1467 really came on later on in the project for me because we actually started studying it because we wanted to use it as a positive control for a lure assay because it's been shown to inhibit lure in another microbial organism. But when I started working with it, we realized, oh, this is actually a good potential anti-malarial compound. And so I added that to my list of compounds of interest. And so aminoacyltyrrhinase synthetases in general have been explored by a lot of different groups for their potential to be drug targets. And this is especially true in prokaryotes because these enzymes in a prokaryotic background is different enough from a eukaryotic background to allow for selectivity. So currently there's a drug, FDA drug called mupirocin that targets isoleucyltyrrhinase synthase, and this is usually used for MRSA treatment. And this idea of using aminoacyltyrrhinase synthetases is also has been entertained in plasmodium and in some other apicomplexans because of the presence of the apicoplast. And the apicoplast, as a lot of you would know, is an organelle that's believed to have been derived from a prokaryotic origin due to endosymbiosis. And so it has its own translation machinery that's more prokaryotic than it's eukaryotic. And so the idea is that if you're targeting the apicoplast, then you're also increasing selectivity for the parasite versus the human host. The con of this, I mean, there are antibiotics like the tetracyclines that are used against plasmodium, against malaria infection disease right now. But the con of those is that they are usually slow acting because when you're hitting the apicoplast, you really don't begin to see the effect of treatment until after 1 or 2 cycles of the parasite, of the red blood cells stage. And so you usually see these antibiotics used in combination to other antimalarial compounds. However, there are some aminoacyltyrrhinase synthase inhibitors that have been identified to target the cytoplasmic compartment of the plasmodium parasite that's selective enough. So for example, this falciparum was published by the Winslow lab in 2012 showing that this compound selectively inhibits lysotyrrhinase synthase in plasmodium compared to, doesn't have any effect, any effect on the human enzyme. So what is Luares? We know it's one of 20 aminoacyltyrrhinase synthases. It belongs to the class 1 tyrrhinase synthase family, and it contains 2 separate active sites, one for the aminoacylation reaction itself and the other for the editing domain. So this editing activity is essential because you have valine and isoleucine that are similar in structure to leucine, and every once in a while you get a misaminoacylation where a valine takes the place of leucine or an isoleucine takes the place of leucine. So this editing activity is essential to increase the fidelity of the protein synthesis process. And this is just kind of like a schematic showing how the reaction happens. And so you have your enzyme with your leucine tRNA, and you use ATP to pretty much generate the leucine that's bound to the tRNA. So one of the first things we did, of course, is to look at the activity of these compounds in vitro and in vivo. So we did IC50 studies, and we found that this 1467 compound has an IC50 of about 0.2 to 0.4 micromolar against the drug-sensitive DD2, drug-sensitive 3D7 and the drug-resistant W2 and DD2. The interesting thing is, I mentioned earlier, this 1466 is the inactive enantiomer for 1467. In vitro, they all seem to have comparable IC50 values. However, when you go in vivo in mice, this is an ED90 that's calculated based on a 4-day parasitemia in the mice after drug treatment. You see that the ED90 values vary very much. So these compounds that are more alike each other have much better ED90s compared to the inactive counterparts. 3661 was had much better activity in vitro and in vivo with an IC50 of about 30 nanomolar and an ED90 of about 0.3. Our collaborator Roland Roland is from the Dominican University, and he was in Uganda, and we were privileged that he was able to test these compounds against isolates in the field. And we saw comparable IC50s. There are a few that are, a few lots higher than the IC50s that we observed in lab. So one of the first things that we did, I should have gone this far to know, but one of the first things that we did was to start selecting for resistance. So what this is supposed to show you is the mode of selection. So because I started working with 3661 much earlier than the 1464, we had more selection with that. So this is a W2 parasite. We selected against W2 and against DD2. And then we selected using two different techniques. So a stepwise technique versus a one-time technique. So this really, we started with 3% parasitemia tail culture at 2% hematocrit in 3-plicate flasks. And then we cultured them in the presence of the IC50 of the compound, of the 3661 compound for about after 43 days. Here we started seeing parasites that were doing as well as the non-treated parasites. And then we saved an aliquot of that and increased the concentration again. And so we did that for 5 generations. And this is supposed to show you the range of the IC50s that we got. And we did the same thing with DD2, but we didn't do as much of a selection compared to the W2. The FIDA lab in Colombia did a different type of selection where they started with a much larger number of parasites, and they did this in DD2 background and incubated them in a larger concentration of compound and just did a one-time selection in 3 different flasks. And for 1464, we followed the same, we did this in a lab, and this was with the same, using the same stepwise drug selection technique. And so while this was going on, we were doing a bunch of other different assays to characterize the mechanism of action for the compound. So one of the first things that we did was to look at stage specificity. A lot of the anti-malarial drugs that are out there tend to be more active against the trophozoite stages, because that's the most metabolically active stage of the parasite. And here you see that the compound selectively inhibits the trophozoite stages more compared to the rings versus the Scheisen stages. And we used chloroquine here as a positive control. When we did the same thing with compound 3661, we saw the same effect where we saw a preferable inhibition of trophozoites compared to Scheisens and compared to rings and Scheisens. So another thing we did was to look at protein synthesis. If you are inhibiting lucinotyronate synthetase, you would expect that protein synthesis would be affected in one way or another. And what we did here, what you're looking at here is incorporation of radiolabeled leucine. We could have used any amino acid really, but we used leucine, radiolabeled leucine. And what you're looking here is a percentage compared to untreated. And here is the x-axis showing you the drug concentrations represented by the fold IC50. Cyclohexamide was used as a positive control. Artemisinin was used as our negative control. And you see here cyclohexamide, a nice dose-dependent inhibition of incorporation. Compound 3661, I would say, had a very, very moderate inhibition. If any, I mean, it was below 100%, but still high up there. However, compound 1467 and 1474, which were the active compounds, the active blue RS compounds, as identified in a different microbial background, both showed very nice dose-dependent inhibition of leucine incorporation. Comparing that to the negative enantiomers, you see that you don't see any inhibition of protein synthesis at all with those. So another thing we did was to then go in and probe directly leucine synthase activity. We didn't have a recombinant enzyme, so we used a crude extract as a source of enzyme. And the way we tested this was to incubate crude extract in the presence of exogenous tRNA, in the presence of drug, and monitor the incorporation of radiolabeled leucine. First, we use tRNA from Cerevisiae. There's been some publications out there talking about species selectivity when it comes to the use of amino acid tRNA synthetases and the tRNAs that they're able to charge. And so the hypothesis is that a eukaryotic enzyme is most likely to tag a eukaryotic tRNA, and a prokaryotic enzyme is most likely to tag a prokaryotic tRNA. So we just kind of try to use a tRNA from a prokaryotic source and a eukaryotic source to see if we can differentiate the activity between the apicoplast lure versus the cytoplasmic lure. So here is using the Cerevisiae tRNA, and you see here that compound 3661 and the negative enantiomers, 1466 and 1478, had no inhibition of incorporation. However, you do see some inhibition of incorporation of leucine in the presence of the two active 1647 and 1474. We did the same thing using E. coli tRNA, and we got the same thing. So I guess the question here is, well, are we detecting, is this detecting the cytoplasmic lure activity while this is detecting the E. coli tRNA activity? We don't know that. What we can say is that there are things, a few things that may be going on. It could be that you have the cytoplasmic enzyme that's able to use both the Cerevisiae tRNA and the E. coli tRNA. Or you could have the E. coli enzyme, the apicoplast enzyme that's able to use both tRNAs, or you could have both of them being detected. We did try using E. coli enzyme to test against Cerevisiae tRNA, and we got activity. So it could, and the whole hypothesis about species selectivity is really not universal. It just depending on the species that's being compared. But what we can say here is that with 1467 and 1474, that we do see an inhibition of incorporation, which we think is affecting the LUARS enzyme. And so at this point, we had finished selecting for resistance to the compounds, and each of the generations was cloned by serial dilutions. And then we prepped those and sequenced them with Joe DeRisi's lab, who also provided training to us for how to analyze the data. And so this is just kind of to show you, I don't want to dwell on this too much, but this is really to just, the point I'm trying to make here is we got a lot of SNPs. But the whole point is you get a lot of SNPs and then you find a way to tease out which SNPs make sense, which ones should you be focusing on. We had a total of about 3,000. And then a lot of those were in non-coding regions, and then you have coding regions, and then you had exons and introns, and then the exons, the SNPs in the exons were disqualified for different reasons. One being if you're a VAR gene, you were probably not a target, and then also the consistency of the SNP at any different position was also considered. But at the end of the day, we got SNPs that were the SNPs of interest that we were looking into were 3 non-synonymous SNPs from each of the different. So at this point, we only sequenced the first and the second generation parasites. The third generation parasites are currently still being sequenced right now. So the 3 genes of interest for each of the different generations are listed here. But for time purposes, I'm going to just focus on this, where we saw SNPs in the cytoplasmic leucine synthetase. We didn't see any SNPs in the apicoplast leucine synthetase. And if you look more directly, these were the SNPs that were found. So what we did was we went back after the whole genome sequencing and we tried to validate the SNPs in the leucine and the LUARS using Sanger sequencing. So a lot of these, so for the ones that have 1 check mark, means we only have data from Sanger, And for the ones that have 2 check marks means we have whole genome sequencing data and standard sequencing data. And you see that all these are the 4 different, these 2 were found in the R1 generation, and this was found in the R3 generation and the R2 generation. And this is the R2 only. And there were no SNPs identified in the parental DD2. And all of these SNPs fall within the editing domain of the LUARS enzyme. And so we went back and we repeated the protein synthesis assays, but this time comparing parental parasites to drug-resistant parasites. And here we're still looking at incorporation of leucine, comparing that to fold IC50 of the parental DD2 now, not the fold IC50 of the resistant parasites. And what you see here is that the parental DD2 and the first resistant generation are still susceptible to inhibition of incorporation by this compound, but the R2 and the R3 resistant lines were resistant to inhibition. And so what this is saying, and this is the beauty of using the step-by-step selection, because you're able to capture what the parasite is doing from going from low resistant levels to high resistant levels. And so this clearly is saying that even though this was a resistant line, the mutation in the LUARS was not essential for resistance. And so it did a bunch of other things until it found whatever that worked better for it. And then another thing that we tried to do was to really try to probe and look into the implications of LUARS in this system. A slam dunk obviously would be to have a recombinant enzyme where you're able to test directly the effect of the compound on enzyme activity. But we couldn't get that. Our attempts to express this enzyme failed. So what we did instead was to do this assay that I'm going to call the Novaline assay. Something similar was done in the 2007 science paper that was published for another benzoxaborole compound. And what this really means is if you have a parasite that has an intact editing activity, the fact is that if you put in this case, Novaline, which is a type of valine, if you put in this amino acid analog into the culture, a parasite with an intact editing activity should be able to cleave it off if it goes in place of leucine. So if you have a good LUARS editing activity, the parasites should not be able to be affected much in the presence of Novaline because they're just going to hydrolyze it off and keep going on. And so what we did was to test parasite growth in the presence of varying concentrations of Novaline, both in the presence and in the absence of leucine. And in the absence of leucine, you would expect that Novaline would have a much more impact on the parasite versus in the presence of leucine. And so if you have an impaired editing activity, then having Novaline is going to be more detrimental to you, the parasite, because it's not able to cleave off the Novaline and have the correct leucine. And so when we did that, it was exactly what we saw. We had a wild type and the first resistant generation were able to do fairly well in the presence of increasing concentrations of Novaline. You're looking here at control parasitemia, and this is an increasing concentration of Novaline. However, the second and the third generation resistant parasites were not able to do that well in the presence of increasing concentrations of Novaline, which means that they're not able to cleave off the Novaline, leading to production of nonsense protein that eventually leads to parasite death. However, this is in the absence of leucine, in the absence of exogenous leucine, because as you all know, the parasite can get leucine from hemoglobin breakdown. So we can't say there is absolutely no leucine in the system. However, when you add exogenous leucine in the RPMI to normal media levels, you see that you rescue even the R2 and the R3 resistant stages that we're not doing very well here, until at a point where you begin to get to a higher concentration, they begin to taper off again. And so the question is, well, you get a compound that has a hit. You identify the target. What exactly are the implications? Because if you are not able to select, if it's not selective for the parasite enzyme and it's hitting the human enzyme, then it's really of no use. And so then the question is, how different is the human URS from the cytoplasmic enzyme in falciparum? And this is really just looking at the editing domain of both the human and the falciparum URS. And these are highlights of where the SNPs were observed. And so what you see, there is a decent amount of homology, sequence identity of 27% and a similarity of 45%. So on a primary sequence level, this is I would say this is conserved to a very reasonable level. And as you can see here, some of these positions where we observed the SNPs in falciparum are also conserved in the human enzyme. So what does this mean? In metazoans in general, URS exists as a big multi-amino acyl tyrranase synthetase complex that comprises of 9 amino acyl tyrranase enzymes total, and 3 other non-synthetase proteins. And so what this may mean is that maybe there are some steric hindrance situations going on where the compound is not able to access the human URS as much as it's able to access the P-falciparum URS, which is a monomer. And we did some CC50 assays comparing the IC50. This is a comparable IC50 assay against human Jurkin cell lines, and we saw that the CC50 is definitely more than 100 micromolar, which compared to the IC50 of parasites of 2 to 400 nanomolar is quite different. So something is definitely going on where the parasite is selectively much more, the compound is able to selectively inhibit the parasite enzyme and not the human enzyme. And so, quick summary for this compound. We know that it's a promising antimalarial drug candidate, and we're still evaluating this compound and other compounds for antimalarial drugs. And we've shown that this compound inhibits the trophozoite stage more than the other stages. We've shown here that a dose-dependent protein synthesis was observed with compounds that were treated, with parasites that were treated with this compound and the relative compound 1467, or rather 1476, sorry. And we did see the same dose-dependent inhibition in LURS, in the LURS assay, crude assay that we did. And parasites also had, the parasites that were selected for resistance, we were able to show SNPs in the cytoplasmic glucosyltyrranase synthetase. And the editing function, we could say that parasites, the resistant parasites had impaired growth in the presence of novaline, which may be suggesting that there's an impairment in the editing function. So to quickly go over to the 3661, we also whole genome sequenced these, and instead of glucosyltyrranase synthetase, we found SNPs in this protein called cleavage and polyadetylation specificity factor. And all of the SNPs, we found a whole lot of SNPs just like we did in the 1467 resistant parasites. But we eventually narrowed them down to these. And the interesting thing is the first resistant line, so far we only sequenced R1, R4, and R5. R2 and R3 are still in process. But R1 didn't have any SNPs in this protein. However, R4 and R5 had these SNPs. And this is the W2 background, and this is the DD2 background that was selected in a stepwise manner in our lab. If you go to the other, these are the parasites that were selected in David Fiddock's lab in Columbia, and we saw the same gene implicated. So here we see the same gene implicated in 2 different parasite backgrounds, selected using 2 different techniques for resistance. So we've been looking into this protein to really understand, okay, well, what does it do? Going in depth, so another thing, what we did, like we did in the previous, with the previous compound, was to then go back and validate this compound, these SNPs again by standard sequencing. And so the interesting thing that was going on, so R2 and R3, we haven't whole genome sequenced them, but we were able to standard sequence the CPSF. And there were no SNPs in both of them. In the first two generations of resistant parasites, we didn't see SNPs. However, when we looked at the PFMDR1 copy number, we went in a W2 background from a copy number of 1 and immediately shot up to 4, between 4 and 5, between these two. And so what's really going on here could be that the parasite, as a first line of defense, increased the PFMDR1 copy number to fight off the parasite pressure. But eventually that wasn't enough, and so it had to start making changes in this gene that it probably wouldn't want to do if it didn't have an option to do that. So in our DD2, we didn't see any variations in the PFMDR1 copy number, most likely because DD2 already has a 3 copy number, of PFMDR1 copy number of 3. And these are the ones that were selected in David Fiddock's lab. And those also had the SNPs, except for the R3, but this could be because this was an uncloned population, so we weren't able to detect the SNPs there. And so what's CPSF? The name, as it indicates, is a cleavage. It's a protein that's involved in cleavage and polyadenylation of the 3' end of pre-mRNA, both in the normal pre-mRNA and in histone pre-mRNA. So the difference between these two is that in normal pre-mRNA processing, you have cleavage and then polyadenylation. However, in the histone situation, you only have cleavage. Histone pre-mRNAs are actually not polyadenylated. And the thing that connects these two different processes is this CPSF73, CPSF100, and this simple kin, which is not shown here. But these 3 proteins are common in both processes. There are 4 different CPSF proteins, and these are all indications of how big their kilodalton values. And so 73 and 100 are very, very highly similar and have been shown to interact with each other in both processes. And so looking more closely into both of them, both of them belong to a class of proteins called the metallo-beta-lactamase hydrolases, and they both share, like I mentioned, very similar tertiary structures. So this is a publication that came out in Nature, showing the crystal structure. This is a schematic of both. So this is CPSF73, and this is CPSF100, and they both have similar structure. However, CPSF100 was found to not have any endonuclease activity because it lacked the required motifs that were necessary for cleavage. However, CPSF73 didn't. So they were able to identify that the actual cleavage that happens in polyadenylation process is due to the CPSF73. So then the question is, when you search on plasmodium, you just get CPSF. It doesn't designate whether it's CPSF73 or CPSF100. So which one is which? There are these 4 different results that come up when you search for CPSF on plasmodium. I was focusing more on these because both of these are actually belong to the metallo-beta-lactamases groups. And so then the question, the SNPs that we identified were the SNP on this one, on chromosome 14. So then the question is this the CPSF73 or is this the CPSF100? So what we know, just based on doing some searches, is that the CPSF14, which is where we found the SNPs, is a 101 kilodalton protein, and it has a 29% sequence identity and 56% similarity to human CPSF73. And it also has a 16% sequence identity and 47% similarity to the CPSF100. The one on chromosome 3 is 118 kilodalton. It has lower sequence identity compared to CPSF73 compared to the one on chromosome 14. And so if you look at the structures, this was gotten from the Sanga website. So this is CPSF73 in humans, and this is CPSF, this is where we found a SNP, the CPSF14, and this is the CPSF on chromosome 3. And you see that they're both characterized by this beta-lactamase domains and this beta-caspase domains. However, the C-terminal is not conserved with the CPSF33. So I think at this point we're leaning towards the fact that this CPSF on chromosome 14 is most likely a CPSF73 because it looks much more like it than the chromosome 3 enzyme. And if you zoom in, if you were to kind of take a look at what's going on the sequence level. So here I've highlighted in yellow, these metallo-beta-lactamases are characterized by 5 different motifs. And so this is motif 1, motif 2, 3, 4, and then these 3 here make up the motif 5. And so what you're seeing here, the reason why CPSF 100 doesn't have an endonuclease activity is because it's not conserved at these very essential motifs. So it's lacking the histidine, for example, here, and the aspartic acid that's necessary here for endonuclease activity. However, you see that all those places are conserved in the 73 compared to the protein where we found at SNPs. So this is a stronger evidence that what the protein that we're looking at is more likely a CPSF73 than a CPSF100. And so the summary for this compound, we still have a lot more work to do on it, but we know we're still trying to look at other related compounds to this compound, the related compounds as potential antimalarial candidates. We do know that it inhibits the trophozoite stage more than the other stages, and we know that we saw SNPs in this CPSF protein, this CPSF gene, across all the different generation resistance that we had. And we saw a very moderate effect on protein synthesis that may be an indication of what's going on the mRNA level if this is in fact targeting the CPSF as we think it is. And we're doing a lot more work on this compound to characterize CPSF as a target by looking at mRNA abundance, comparing that in treated compounds versus untreated compounds. And we're also expressing the recombinant enzyme for this to see what's going on. And also the David Fidok lab in Columbia is looking into a zinc nuclease, a zinc finger nuclease assay to characterize the different SNPs that we found on effect on drug, on the enzyme activity. And so the conclusions in general is that drug-resistant selection is a powerful technique to establish mechanism of action for novel compounds, and that leucotyrosine synthase is a target for the compound 1467, and we're looking more into these kinds of compounds for optimization. And LURS is a promising target for antimalarial drug discovery, and we do have strong genetic evidence that CPSF could be a target for 3661. And I would like to say thank you to Phil and the members of the Rosenthal lab, most especially Duke, does a lot of the parasite work and a lot of the in vivo studies with URI, and Roland's lab for all their collaboration on the field. And this is the group at Anacor. Shen is the one doing the enzyme expression work now on CPSF. And Joe's lab for helping us with the training and the sequencing for the parasites. And the Fidok lab, and our funding comes from NIH and MNB. That's it.
Questions for?
Yeah. So I have 2 questions. So all of the resistance that you showed was in culture with falciparum?
Yes.
Could you show any development of resistance with your Bergii model? That's the first question. And then the second question is, have you tried, there are a lot of software programs where you can actually predict whether or not amino acid changes would be predicted to be pathogenic or, potentially damaging to protein function. Have you tried any of those?
So I'm going to tackle the second question first. We are collaborating with Stephen Cusack on the leucotyrosine synthase enzyme. He's done some work actually. Some preliminary work shows that the SNPs that were identified in the first resistant generations were actually not essential enzyme activity in general. So it wasn't surprising when we saw that generation didn't really look that much different from the parental strain. We haven't done any of that for CPSF yet, but that's something we're definitely gonna.
You can, I mean, these are freeware. I mean, you can do this online yourself and actually use some of these analysis programs to look at whether or not these predicted changes in amino acid and non-synonymous substitutions would be deleterious to protein function.
Well, yeah, we can definitely look into that. I haven't looked into that. For the in vivo model, what we do, I mean, that would require, I guess, treating the parasites, the mice with the compounds for a long period of time to see what happens to the P. burgeriae. We haven't really done that kind of work, but that would be something to look into.
I have somewhat of a naive question, I guess. Just looking at, it seems like you got an awful lot of SNPs, but my reference is Cerevisiae, where when you would do, if you were to do a similar type experiment, you wouldn't get, you'd get orders of magnitude fewer SNPs. So I'm wondering, is that normal for falciparum, or is it?
That's very normal. It's 80% ET rich, and so that means the energy required to make genetic changes is much higher, is much lower. I mean, you have an AT bond that's 2 versus GC that's 3 bonds, and so falciparum especially, one of the reasons why it's been known to mount resistance against everything is it doesn't seem to cost it that much to make changes on a genetic level, and so that's not surprising that we see that.
And just out of curiosity, so what do you use as a reference for your SNPs? Do you use the parental, or do you? What do you use?
We use the parental, yeah, the 3D7 or plasmodium.
Any other questions?
So one of your early slides, you had, I can't remember the numbers, you had 2 enantiomers, both of which had similar activity in the in vitro assay, but very different activities in the P. burgeriae model. Any idea why that was?
That's a good question. We kind of try to think about what may be going on there. One is definitely the negative enantiomer in the microbial background that in the unpublished work that's been seen. In that system, it had a poor in vitro and in vivo numbers, and so in our case we saw poor in vivo activity but very good in vitro activity, and that may be saying something about the access, how well is the compound getting into the cell in vivo background. It could be that, or it could be that there may be some off-target that may be involved, where the negative enantiomer is doing something else when it's able to get into the cell that's unrelated to low arrest activation.
To kind of piggyback off that question, at what point in this process do you look to see how well your drugs are sticking around in vivo? So when you're doing those P. burgeriae treatments, do you look to see how quickly they're being metabolized? Because, I mean, the thing I would think is if you're getting different levels of activity, the easiest thing that would occur to me is that they're just getting metabolized more quickly.
So we haven't looked at PK in using for this compound. For 3661 we do, and we have information on what's going on. But I couldn't really say much about what's going on as far as the drug levels in mice in vivo for that compound, because we haven't looked at PK levels yet. But that's definitely something to keep in mind. It's the clearance level of the two of them to see who's lingering around more than the other.
I have a couple questions. So first of all, I don't quite understand what's the model for the CFRS and how does that provide the drug resistance?
So you mean the CPSF?
CPSF, yeah.
What's the model?
Yeah, how does it provide the drug resistance? What is it doing?
We don't know. So that's definitely something that we want to be looking into. We don't know exactly how it's inhibited. You're asking how the compound is interacting with the enzyme, right? To inhibit its activity?
Yeah, how is it, exactly, so how is it providing the drug resistance?
Right, we don't know how, I mean, the whole point is if this compound is inhibiting that target, for example, it's targeting the target, if it's inhibiting, if the compound is inhibiting CPSF, then chances are it's inhibited in such a way that those mutations are, obviating the pressure. I'm not sure if that makes, but we still have to do a whole lot more work in that area to find out how exactly it's interacting with the enzymes.
And then how do you actually know that it actually isn't interacting with that particular gene? Because you're looking at a resistance, and so you're not actually sure that, like even the blue RS case, it might be acting on something else as the product that the blue RS is acting on another gene, right?
Right.
So you don't know per se if it's acting exactly on that gene.
So that's why in the conclusion, we're not making a strong conclusion that CPSF is a target because we still need to do more work. Usually when in resistant, what you usually get is usually a snip in a pump that gets rid of the compound. But usually what you see, at least from what we see, we can maybe make a conclusion that it's affecting the polyadenylation process. But whether or not, like you saw in that picture, it may not be CPSF73. It may be something else that interacts with CPSF73 or CPSF. So we still have to tease all that out. That's why we're not making a strong conclusion that that's the main target of this one.
Okay, if there's no further questions, thanks, Barry.